So the topic of my today's talk is Helminth control in sheep and goat with the improved or you can say sustainable approaches or you can say alternative approaches to control the helminths of the small ruminant sheep and goat. Now we all know that nowadays there are newly emerging threats to continue successful small ruminant population, and it has led to the failure of chemotherapeutics due to widespread development of anthelmintic resistance. And nowadays what we have seen that there is changing of people attitudes towards more of animal welfare, food preferences and safety. It signifies the impact in which the small animals they should be managed and their parasites should be controlled. Thus, now it has become a recognized fact that we should switch from more hard methods away from chemotherapeutics to something which are more akin to the parasite management, and that is to live with worms. Now, to start my topic exactly, let us little see about the background of various helminths, particularly the nematodes, which infect our small ruminants, the sheep and goat. So most of them they inhabits the abomasum of sheep and goat, like the most hemophagous or you can say most voracious bloodsucker, the Haemonchus contortus. It inhabits the abomasum of sheep and goat, and you can say that each worm it sucks about 0.05 ml of blood per day. It causes anemia in the animal, and you can very well during the post-mortem examination you can see the red bite marks of the parasite. It is commonly known as barber pole worm, or you can say the brown stomach worm. It is also known as bankrupt worm because it lives on the all the nourish it utilizes all the nourishment given to the animal. And then in the series we have Mesostoceros digitatus. Morphologically it is similar to that of Haemonchus contortus. Then Tila dorsagia circumcincta. This is also an important parasite of sheep and goat residing in their abomasum. Then Trichostrongylus axii. It resides in the abomasum of sheep and goat. And it's another species Colubriformis. It is found in the small intestine of this small ruminant sheep and goat. Then we also have certain voracious bloodsuckers like Bunostomum, Phlebotomum. It resides in the small intestine of sheep and goat, and it sucks the blood. It feeds on the blood of the animals. Then we have Cuperia and also Esophagostomum columbianum. It causes pimply gut, or you can say naughty gut. Then we have also Trichuris ovis residing in the cecum and colon. Now, most of the parasites they are transmitted through their per oral ingestion, or you can say through ingestion of contaminated feed and water containing the parasite or their stages. Except you can say Strongyloides papilosus, where the major route of transmission is percutaneous route through skin penetration. But also per-oral infection can be there. Then some of the parasites, they can be transmitted transcholesterally, or you can say transplacentally, like Toxocara vitulorum, and also the hookworms. Now coming to our topic of interest, that is the emergence of enthelmintic resistance, which has led to switch to some better, some sustainable measures of control. Now, unfortunately, the enthelmintics, they are generally not used in a sophisticated manner. They are sometimes overused, misused, or applied incorrectly. As a result, there is widespread development of enthelmintic resistance. And moreover, people have become more aware about the animal welfare. They have some emotions attached with the animal, and people are going for organic everywhere. Their mindset has been changed from this drugs residues to something organic. They're going for organic farming, they're going for organic meat, they're going for organic pulses. So to start with, we have a basket of options approach. This has been adopted for particularly Haemonchus contortus infection in small ruminants. These approaches are practicable, affordable, and easily available not only to the commercial producer but also to the resource poor farmer. We have number of approaches under this basket of option approach. So first is grazing management. Here comes the importance of zero grazing. So here what we do, rather we allowing the animal for open grazing, we prefer to offer them cut and carry herbage, and particularly we stall fed them rather than allowing them to graze in the open. So animals are stall fed here with treated forage or silage, and this prevents the growth of the parasite. Next is rotational grazing. Here, the pasture is divided into small paddocks, and more animals are allowed frequently to graze on different paddocks. So here, each paddock will have a rest period, which is enough to break the life cycle of the parasite. So it has been seen if we allow smaller periods of grazing and give more period of rest, so it will prevent the parasitic transmission in a broader way. Then another method is mixed grazing, or you can say co-grazing, where grazing different species of animal together like cattle and sheep, it can help to control the nematode due to their different grazing habits, disrupting their distribution. So adopting such basket of options approach, such newer approaches, we have in Asia the Sparsi network, which promotes the use of such approaches. Basically, it is a combination of these approaches together to prevent the worm population in a broader way. Now we can divide the control of Helminth population in small ruminant of sheep and goat broadly into two categories: the suprapopulation control and the infrapopulation control. Suprapopulation control is control of parasitic stages in the pasture, or you can say outside the animal. Infrapopulation control is control of parasites within the animal. So here we have number of factors like grazing management, using predaceous fungi, and here in infrapopulation we have immunonutrition, genetic selection, use of bioactive forages, then use of copper wire particles, and use of targeted selective treatment. Now, grazing management. So, the goal is to provide a pasture which is free from parasite for grazing, is covered under grazing management. So, it has been seen that a pasture which has not been grazed for about 6 months in case of sheep or goat, and in case of their young ones, and in cold weather, and 3 months during hot or dry weather, is found optimum for grazing of the animals. Then coming to nematophagous fungi. So the most popularly is Dudingtonia flagrans. It was first discovered in New Zealand. So here the chlamydospores, they are basically the ovicidal. They are fed to the animal, and they can survive the passage throughout the gut. As such, the spores are excreted in the feces, and what they do, they will destroy the larval stages, or you can say developing stages of the parasite through their specialized trapping mechanism. And this has also been marketed or commercialized products are available in the market under the name. We'll cover that slide later, but here we have seen that there is reduction in this, using such spores have reduced the fecal account to more than 60%. And another fungi is Arthrobotrys oligospora. It is capable of reducing the infection of Strongyloides papillosus parasite in a long way. So this Arthrobotrys oligospora, it has also been evaluated in the control of hookworm larvae, the bunostomum, and it has given promising results. So this fungus, Dudingtonia, it's being marketed under this name bioverma. So it is easily available in the market and it reduces the fecal account in a greater way. Now coming to some of the botanical dewormers like teniferous plants like chicory, allium, acacia, pumpkin seeds, calendula, turmeric, garlic and mustard seeds. So these are also proved to be very effective in controlling the worm population. This chicory is grown in the state of this Uttar Pradesh and also in Gujarat, and it is perennial herb, that is it is available round the year, and it reduces the larval survival on the pastures. Then this garlic, this have a bioactive compound known as allicin, which have anti-parasitic properties when included in the diet of the animal. Then this mustard seeds are easily available at home also. Papaya, it also has a peristaltic effect which reduces the worm burden. These dung beetles and earthworms, they can help to reduce the infective stages on a pasture, and it has been seen that the use of dung beetle reduces up to 15% fecal account and earthworms up to 50%. Then we have also some nematophagous mites like Myostigmatid has recently been identified to be a great predator of sheep gastrointestinal larvae. Now, in the series, we have immunonutrition. In this immunonutrition, if we give a balanced diet, we offer a balanced diet, balanced ration to the animal, so it will improve the immunity of the animal to fight the parasitic infection or infestation. So balanced diet, okay, incorporation not only includes the carbohydrate, protein, but also the minerals, also the trace elements. It supports the optimum function of the immune system and improves the overall health of the animal, making the animal more resistant to the parasite. So it has been seen that supplementation with protein sources like soybean meal, fish meal, it reduces the fecal account and worm burden by 60%. But as we all know that protein are little expensive, so we can switch to use of some low cost supplements like molasses, urea block. It improves both the resilience and resistance among the animals. Now, the research have shown that this gastrointestinal nematode, they will increase the demand of protein, or you can say amino acid, by the infected animals. So we should incorporate optimum proteins in the ration of the animal. And it has been seen that supplementation with phosphorus is also important in preventing the establishment of the worm. And cobalt deficiency has also found to be affecting the immunity against the gastrointestinal nematode. It will make animal more prone to the parasitic infestation. So if we incorporate all these nutrients in their required amount in the diet of the animal, so it will stimulate their immune response, their ability to fight with the infection, and their productivity, their production will ultimately increase. Now coming to another approach for the long-term viability, this breeding for resistance is one of the best approach, as the genes for resistance can be fixed in the host population. So it has been found that Gulf Coast native and St. Croix breeds are more resistant compared to Suffolk, Dorset, Rambouillet breeds of sheep. And this Garrol sheep is resistant to Haemonchus contortus infection. It will show less FEC, fecal account, compared to other breeds of sheep. So this Garrol breed and rest Maasai breeds of sheep, they are resistant to Haemonchus contortus infection. And amongst the goat, we have myotonic breeds and Spanish breeds, which are resistant to the infection. And one more breed of goat, that is Boer breed of goat, it is also found to be resistant. Now, use of copper oxide wire particle, it is considered to be the best short-term approach available. Here, the copper particles, they are released and lodged in the abomasum of the animal. So we have seen that most of the parasites, they are residing in the abomasum of the animal, where these copper particles can exhibit anti-parasitic effects. And this have also been marketed commercially available in the market. So these copper sure boluses are available for goat, which are marketed by Animax Vet. Now, use of bioactive forages. So ethno-veterinary medicine using a variety of plant products have been used for many centuries. So here, plant enzymes such as cysteine proteinases, secondary metabolites such as alkaloids, glycosides, tannins, they have anti-parasitic properties. The condensed tannins, Lespedeza cuneta plant, they have high level of condensed tannins and it exhibits both direct as well as indirect effect on the parasite. Now, direct toxic effects include that this condensed tannins, they will bind to the cuticle of the nematode and it will cause the death of the nematode. It will change the abomasal environment and that favors the expulsion of the parasite, like self-cure phenomena, which is exhibited by Haemonchus contortus, Trichostrongylus, where the adult worm population, they are automatically expelled from the gut of the animal. It provides immunity. So we call that self-cure phenomena, which is exhibited by sheep and goat against Haemonchus contortus and also against Trichostrongylus. So they work similarly, that they will change the environment of the abomasum and will favor the expulsion of the parasite through the gut into the feces. And it will also reduce the fecundity of female worms, the ability of the female worms to give rise to eggs and then to larvae, it will be reduced instantly. Then indirect toxic effects include that these condensed tannins, they will bind to proteins and will protect these proteins from human microbial degradation. So ultimately what they are doing, they are improving the availability of proteins. So certain bioactive forages like bird's foot trefoil, scientifically known as Lotus corniculatus, it is found in parts of India, particularly the Himalayan regions. This is used commonly as a forage crop for livestock and it is good because it has high content of proteins. Then another species is Sericea lespedeza. It is the, in Hindi we call it palash. Okay? And this is the name for pea or bean family. It decreases the larval development and worm burden in sheep and goats. Now, another is the managemental strategies that employ refugia based methods. So, it includes targeted time whole flock treatment and targeted selective treatment. Basically, anthelmintic resistance is due to indiscriminate usage of drug as and when. We often go for mass deworming. Rather, we should switch to targeted selective treatment. Means we should target only those animals which really needs treatment rather than offering it to entire population or entire herd of the animals. Now, this term refugia, it refers to individual in a given population of a parasite that escapes to any control method applied. Hence, it is available to dilute both the progeny of a resistant parasite and to mate with the resistant worms. So therefore, this maintenance of this refugia is must if we want to keep our flock free from enthelmintic resistance. This refugia is very important in nowadays people are focusing on this refugia managemental strategies rather than mass deworming. So what are the potential markers? On what basis we will target our animal population which actually need the treatment? So we have certain pathophysiological indicators, like in case of hematophagous parasites, like your Haemonchus, like hookworms, bunostomum. So their anemia. Anemia is one of the clinical signs which will be exhibited by the animal. So depending upon the level of anemia, we can ascertain that whether that animal require the treatment or not. So it is an idea. It has been around for a long time. It's not an idea which is emerged nowadays, but it was an idea which was long back to target or to treat only those animals which actually require the treatment. Normally, parasites are highly over-dispersed. It is 20 to 30% of animals that actually harbor the worms. It's not only 100% will animal will harbor the worms. So we have a method known as Farmer chart, which is from a Pfaffer-Mylan chart, which was devised in South Africa. So depending upon this chart, the clinician or not only the clinician but a normal a common farmer, a field veterinarian, okay, which is not a very sophisticated technique, they can determine, they can ascertain whether their animal requires actual the treatment or not. So basically this pharmacha, it assesses the color of the conjunctival mucosal membrane on a 5-point scale, and they estimate the degree of anemia caused by Haemonchus pontotus. So this level 4 and 5, if the anemia is of level 4 and 5, then we should go for the treatment of the animals. So what are the advantages associated with this? We can treat the sheep side, means instantaneous. We don't need to wait for some lab results or for an expertise to come in. Okay, it's a very instant test, low cost, and it also require low level of expertise. So, in the study of Swarnkar and Singh, according to them, that we should treat the adult when they are in the score, they fall in the score of 4 and 5, and in case of lambs and kids, when they fall in the category 3, 4, and 5. Now, in case of non-hematophagous worms, we use this BOTCON, body condition scoring, to assess the requirement of the anthelmintics or any other treatment per se. Another method is DAG score. DAG score, it is a subjective measurement of the level of breech fecal soiling, and it is applicable for lambs. So depending upon the extension of the soiling of the breech area, we will ascertain whether that animal really wants a treatment or not. Now, some of the parasitological indicators, like I'm from the beginning, I'm talking about FEC, fecal account. So, depending upon fecal account, which is a quantitative measurement of the eggs, oocyst, or cyst of the parasite found in the feces. So, depending upon FEC per gram of feces, how much intensity of eggs or their cyst or their oocyst, we go for the treatment. Then another are performance based indicators like milk production, their live weight gain, then using automated weighing systems. Then this automatic weighing system, this is you can say an advanced technique which was used in Australia, and it can process around hundreds of animal per hour. It's electronic drafting gates linked to database. They direct the animals to different pens according to their body weight, and then based on their body weights, they will list under that they require the treatment or no. Then devising a deworming schedule, like we should not go for indiscriminate use of anthelmintics for the treatment, for the control of the parasites, depending upon their epidemiology of that area, depending upon the complete epidemiological study of that particular parasite, we should devise an appropriate deworming schedule, which is you can say is a zone specific, or you can say a climate specific. We can devise it, and accordingly we should go for the drenching or treatment. So this is a deworming schedule applicable for the northern zone. First deworming for strong guiles should be done in late March, or you can say early April, with the benzimidazoles or levamisole, because we assume that in February, because of winters, okay, there are presence of overwintered larvae in the pastures, or you can say there is periparturient rice. So to control them, to prevent the animal from getting exposed to such increased infection, we should deworm them well in advance, somewhere in when the winters are just switching to summers, okay, in early April or last March, when the winters are ending. Then second deworming for strongyle and ectoparasite should be scheduled somewhere in July or August, in the monsoon season, rainy season, because there is increased temperature as well as increased humidity, which is optimum for the growth of the parasite or their other stages. Then the third deworming, particularly for fasciola in hills and amphistomes in irrigated areas like plains, it should be scheduled somewhere in post monsoon season, you can say November or December. There the population of their vector snail, okay, to control that vector population is also important. So for the control of snail, we can use various molexicides, and also biological control, we can use ducks. Then last is prophylactic measures, immunomodulation. Immunomodulation, or you can say in late terms, vaccination. So a number of anti-parasitic vaccines have been developed, but compared to the protozoans, we have fewer vaccines oriented for Helminth parasites because of number of reasons. As they are multicellular, protozoa are unicellular. To culture them in laboratory is difficult task, and moreover, we have to target the vaccines for different stages of the parasite. We know they have got a complex life cycle. So to maintain a multicellular organism in lab conditions is difficult compared to that of unicellular protozoan parasites. But still we have few vaccines, like for lungworm vaccine, Dictyocolus filarii. Against that we have difil. But now the use have been I think it is no more in use nowadays. It was the only Helminth against which there was a commercially available vaccine. It was a irradiated vaccine which was used to be manufactured in IVRI Bareilly, but now it's no longer being used. Then against Haemonchus contortus, various studies are going on, and there are certain gut membrane proteins which are hidden. They're associated with the gut of the parasites. So various molecules are there like H11 and HGAL, then cysteine proteases, and some of the commercialized vaccines are also available, like a native gut antigen based vaccine. It was launched for the control of hemoptysis in sheep in Australia by the name Barbervax, or you can say Wormvax in Australia, and in South Africa as Wirevax. So work is going on. Most of the candidates, they are limited to the laboratory. It is difficulty to commercialize them. Like one of the tinea ovis vaccine, it is still not commercialized, but intensive work is going on. Now to this remote sensing and geographical information system, it's a novel method of control, or you can say to achieve maximum efficacy of a novel control method is highly dependent on understanding the epidemiology of the parasite. So rather quoting this remote sensing and geographical information system as a method of control, you can say it's a cost effective approach for designing and monitoring the control programs at the realistic scale. Basically, these are the new tools to generate epidemiological data. Epidemiological data not only tells you about the parasite, but its complete life cycle, the vectors involved. Okay, what are the predisposing factors associated with it? Then, in what zones, in what areas, in what regions, what climatic condition the parasitic infection is common. So, all will be generated under epidemiological data. So this approaches remote sensing and geographical information system. It has been attempted for fasciolosis, schistosomosis, filariasis, and the study by Sangwan in the region of Haryana, they have prepared risk map based on these two approaches of snail-borne diseases. So they have found that snail, they are the vectors for most of the trematode parasites. The trematodes, they have their indirect life cycle, and there the first intermediate host are exclusively the snails. So they what they found that snail population is more in areas where there is muddy soil and in proximity of the canals, where the canals are there which will provide them humidity, muddy soil, proper shelter. There the population of snail are more. So here this fasciola, they're transmitted by limnia species. So depending upon them, they have created the risk map, and then they have given forecasting of that particular parasitic diseases, and we can go for a proper anthelmintic approach along with the other sustainable methods of control to prevent such diseases. Now coming to the conclusion, in the context of Helminth control, the mindset of suppression needs to be replaced by management of parasite to maintain the long-term profitable livestock production, particularly for the small ruminants. So here we have to replace the term of suppression with management. We should try living with worms. Then existing effective chemical groups like anthelmintics need to be carefully husbanded, and non-chemotherapeutic methods, they need to be adopted along with the chemotherapeutic methods in a in a proper manner. So lastly, I would like to conclude my today's talk with that overall, no one approach is alone the answer. Okay, approaches that are integrated are necessary. Thank you, sir.